Okay, in this video, we'll be going over fatty acid synthesis. Uh, Basically, what fatty acid synthesis is, it's the storing of acetyl-CoA into different fatty acids, and then they can be modified after. So this is the opposite of what beta oxidation is. Beta oxidation is the breaking down of fatty acids, and then fatty acid synthesis is just the storing of fatty acids for later use when we have high energy. So this is done when the cell is in a high energy state. Basically, when you're in a high energy state, you store this acetyl-CoA as fatty acids, and then when the cell is in a low energy state, you're able to break it down later for different uses. Okay, so this it starts with citrate, and it happens in the cytoplasm. So we'll see here citrate made in TCA inside the mitochondria. It's exported out into the cytosol, and then we have a few reactions. We get acetyl-CoA in the cytosol, and then we make fatty acid. So this is linked to both the TCA cycle and glycolysis. So I'm just going to briefly mention this. So we have glucose. This enters the cell. This goes into glucose 6-phosphate. So this is your normal first step in glycolysis by hexokinase or glucokinase if you're in the liver. You make pyruvate. The reason why we need to go in the mitochondria is because pyruvate dehydrogenase complex only exists in the mitochondria, so we're unable to go directly from pyruvate to acetyl-CoA in the cytosol. So we need to first send pyruvate in mitochondria, and then we run pyruvate dehydrogenase complex to make acetyl-CoA. So this is normal. This is the normal stuff we went over at the beginning of the semester. And then we have citrate synthase, takes acetyl-CoA and oxaloacetate, we make it to citrate, and then citrate is exported. The reason why we need to convert acetyl-CoA to citrate and we can't just go from acetyl-CoA right in the cytosol right away is because acetyl-CoA cannot cross the mitochondrial membrane. It doesn't have a transporter. So we need to convert it to citrate first. And then citrate exits into the cytosol. And then we have this acetyl-CoA, the enzyme that makes acetyl-CoA and oxaloacetate, so it's kind of the opposite of citrate synthase, and then acetyl-CoA starts fatty acid synthesis. So that's kind of just a general overview of what this pathway is about. Okay, so after we go into cytosol, we make acetyl-CoA, we go citrate to acetyl-CoA. We'll see that acetyl-CoA is starting to connect a lot of pathways. So when we go over beta oxidation, we'll see that beta oxidation produces acetyl-CoA. We also see that acetyl-CoA is the starting point for TCA cycle, and it can also be involved in amino acid synthesis. What else do I have here? I think that's all. But yeah, it's think of it as like the currency of the cell. It's involved in a lot of different pathways, so it's a way for the cell to be able to regulate all these different pathways together. Okay, so whenever citrate is exported to the cytosol, it's important to know that this is linked to fatty acid synthesis. So whenever the cell exports citrate outside of the mitochondria, the cell is ready to start fatty acid synthesis. From there, acetyl-CoA is made in the cytosol, and then we can start. And like I said, acetyl-CoA is linked to multiple pathways in the cell. Okay, so let's discuss a little bit about the difference between fatty acid synthesis and fatty acid degradation. So this is the same as beta oxidation. So it can be called different ways. And the reason it's called beta oxidation is because you're oxidizing a beta carbon. And when you do that, you break down fatty acids. So it can be called beta oxidation or fatty acid degradation. Okay. So fatty acid synthesis, this happens in the cytosol, and then beta oxidation happens in the mitochondria. This is very important. This is one of the examples of regulation by location in the cell. Because these two pathways share very similar chemistry, if we run them in the same organelle, they'll just create a fugal cycle, and the cell will lose a lot of energy in the process, because then we'll just be making fatty acids and then breaking it down, So the one way the cell regulates between these two pathways is by location. So fatty acid synthesis occurs in the cytosol, and then degradation happens in the mitochondria. Fatty acid degradation uses CoA carrier protein. We'll talk about all, and then it has separate enzymes. It uses NAD, FAD, and it starts with acetyl-CoA. So we'll talk more about this. I'll have a specific video for beta oxidation in specific. This video is mainly for fatty acid synthesis. Okay, so the carrier for fatty acid synthesis is acyl carrier protein, which we'll talk about and how it's important for the enzyme fatty acid synthase. So we'll discuss all of that. It uses two different enzymes. It uses an enzyme called ACC, and then we use a different enzyme afterward called fatty acid synthase. And that enzyme is a big complex enzyme. It's called the metabolome. It's a multi-enzyme complex, and it pretty much takes acetyl-CoA and malonyl-CoA and it turns it into one big fatty acid. So it's one big enzyme that does that, so that's why it has the complex here. We also need a redox agent. We use an oxidizing agent for beta oxidation, hence the name beta oxidation, and then we use a redox agent in fatty acid synthesis, and the cell generally uses NADPH. So if you think about other pathways that we went over, you'll remember that in the oxidative phase of the pentose phosphate pathway, we make NADPH. Usually when the cell runs the oxidative phase, it's because the cell wants to store acetyl-CoA as fatty acids. So the oxidative phase of the pentose phosphate pathway and fatty acid synthesis usually run together because fatty acid synthesis needs NADPH to use as a reducing agent. And our input is malonyl-CoA for this. Okay. So, like I said, fatty acid synthesis, glycolysis, and TCA are all regulated together. And then the control point for fatty acid synthesis is, so this is the enzyme I talked about, ACC, acetyl-CoA carboxylase, which we'll talk about in more specific details in later slides. Okay, so let's talk more about regulation. So we said it can be regulated by location first. Let's compare the difference between glucose and when it's stored as glycogen and acetyl-CoA when it's stored as fatty acid synthesis. Okay, so they both occur when the cell is in a high energy state. So we can store glucose as glycogen when in a high energy state, and then we can store acetyl-CoA as fatty acids when we're also in a high energy state. The difference will depend on what the tissue needs. So we mentioned in glycogen metabolism that glucose is stored as glycogen when the cell needs a rapid source of energy in the future. So this happens mainly, so an example of that would be like the muscles, because the muscles, when they're contracting, they need to break down their storage source as quickly as possible. So the muscles tend to store their energy as glycogen because they're able to break that glycogen really fast, and then they get rapid access to energy. Lipid synthesis is a little different. Oh, and this happens because you're able to break down with water. Which, if you're confused about that, go watch the glycogen breakdown, and it explains why we use water to break it down. Okay, lipid synthesis is a little different. It's mainly for long-term storage. So the muscle doesn't use that because the muscle requires rapid access to energy. The tissues that use this are usually adipose tissue and liver tissue. So adipose tissue and liver tissue usually are your main two tissues that will use fatty acids for storage. Other tissues use it as well, but those are the main ones we'll talk about. Now this is glucose, and then this would be 3 fatty acids. So one of these is 1 fatty acid each. And the reason they look different is because they can be modified. So you can add double bonds, you can elongate them, make them longer. And then when you have 3 fatty acids linked together, so this is a glycerol molecule, you get your tag, which is a triacyl glycerol molecule. So when we have 3 fatty acids linked together, you get a tag, and it's stored as tags. That makes sense? And then what we'll talk about is how we make one fatty acid in this lecture. But then after that, 3 fatty acids come together, 1 glycerol comes, and then you make your tag. And then tag is stored. Okay, so let's look at the key enzymes and the overall reaction. So the key enzymes, so fatty acid synthesis uses 2 enzymes, acetyl-CoA carboxylase and fatty acid synthase. So we said this is a metabolome, which means it's a complex of different enzymes. So it runs 7 different reactions, which we'll talk about. Okay. So in order for the cell to run fatty acid synthesis, so this is the overall reaction, to make 1 palmidate. So this is, I think palmidate is 14 carbons, I want to say, or maybe 16. I can't remember. But we use 18, it's a 14 carbon or a 16 carbon fatty acid. I can't remember on the top of my head. But we use 8 acetyl-CoAs, I think it's 16, I'm pretty sure it's 16. We use 7 ATPs, we use 14 NADPH, and then we use a bunch of hydrogens. So if we go this direction, we do fatty acid synthesis. And if we go the other direction, we'll do fatty acid degradation or beta oxidation. So we'll see that we require a lot of ATP and NADPH to run fatty acid synthesis. So this will come from the oxidative phase of the pentose phosphate pathway. Okay, the ATP can come from a lot of different sources. So this can come from glycolysis. It can come from the electron transport chain if that's running. So the cell will need to run those 2 things, or an equivalent of those 2 things to supply the ATP and the NADPH for fatty acid synthesis to run. So so we said that fatty acid synthesis happens in the cytosol, but it actually starts in the mitochondrial matrix. So because our start point is citrate. So when we think about where citrate comes from the TCA, so we also need lots of TCA activity. So you see that in order for the cell to store acetyl-CoA as fatty acids, we'll need to have a lot of citrate activity for TCA. We'll need a lot of citrate, and that means a lot of TCA activity. We'll need a lot of ATP that comes from glycolysis, and then we'll need a bunch of NADPH, which comes from the oxidative phase of the pentose phosphate pathway. Okay, so this is our start point, citrate. It happens in the cytosol, but because citrate is in the matrix made by TCA, our start point is in the matrix. Whenever we have a lot of citrate, that's one way the cell signals that it has high energy, and then it's exported, and then we make our fatty acids in the cytosol. So usually, whenever citrate is exported, the cell knows it's going to do fatty acid synthesis. The reason we convert to citrate, like I mentioned, is because acetyl-CoA can't be exported because it can't cross the membrane. And we'll talk about the shuttle that the cell, not a shuttle, it's a transporter that the cell uses to accomplish this task. Okay, so let's talk about when you have high blood glucose levels and high ATP levels. How does that translate into fatty acid synthesis? Okay, so those are different tissues. They each have their own GLUT transporter. GLUT transporter just transports glucose from the blood into the cell. So the liver uses GLUT1 and 2, brain uses GLUT1 and 3, adipose tissue uses GLUT1 and 4, and then muscle uses GLUT1 and 4. GLUT2 has the lowest affinity, and GLUT3 has the highest affinity. This kind of makes sense because we said the brain runs mainly on glycolysis, and it uses like 20%, 5% of the glucose your body ingests, something like that. I don't remember the number exactly. So it needs to have a really high affinity GLUT transporter, so it takes most of the glucose in the blood for the brain to run. The liver is the organ that regulates glucose blood levels, so that's why it has the low affinity GLUT transporter, because the liver only wants to take glucose out of the blood when you have high blood glucose levels. If you have medium glucose blood levels, you want to send that glucose to other tissues and not the liver. And if you have low glucose blood levels, you want the liver to send glucose out of the liver into the blood to give it to other organs. So that's why the liver runs a low affinity GLUT transporter. It's because its main function is to regulate blood glucose levels. Okay, so like I said, the liver and the adipose tissue will usually shunt that glucose that comes from in the blood into the cell into fatty acid synthesis. So let's talk about how this happens. So we have glucose inside the cell, and then we make G6P, and then we make pyruvate, and then pyruvate can then be converted into acetyl-CoA, and then acetyl-CoA can be converted into citrate. Okay, so this glucose, so when we convert from glucose all the way down to pyruvate, we'll make the ATP that the cell needs. Okay, the glucose 6-phosphate can go into the oxidative phase of the pentose phosphate pathway, and then this will give us NADPH, which we said we need a lot of as well. And then our citrate can be our start point for fatty acid synthesis. So you see how everything connects together. Whenever we have a high blood glucose level that's able to be shunted into the oxidative phase, we'll make NADPH, and then we'll be able to make ATP in the process of making pyruvate, and then our pyruvate can be converted into citrate. Then citrate is exported, and then you start making your fatty acids. So that's how the cell makes fatty acids when it has high blood glucose levels. So that's why it's regulated with glycolysis in TCA as well. And then glucose can go into the oxidative phase to make NADPH. And then usually when citrate is exported, this is coupled to NADPH production. So look at a different picture of this. It's better than my drawing, of course. So this is our glucose 6-phosphate. So I think they just forgot to add the glucose to glucose 6-phosphate. So we have high blood glucose levels. It enters the cell. It's converted to glucose 6-phosphate. Glucose 6-phosphate can go into the pentose phosphate pathways. So this would make us NADPH. And then glucose 6-phosphate can continue into glycolysis, make pyruvate. Pyruvate enters the mitochondria. We run the pyruvate dehydrogenase complex to make acetyl-CoA. Acetyl-CoA with oxaloacetate will make citrate. And then citrate can continue TCA cycle to make more ATP if it runs this way. And then some of it, because we have a bunch of citrate, because we have high glucose levels, can be exported, and then we go citrate, the acetyl-CoA, and then we make fatty acids. Does that make sense? So this is how it all connects together when we have high blood glucose levels. Okay, so let's talk about how citrate is exported from the mitochondria into the cytosol. So let's start with this. So we have pyruvate. It enters the cell from glycolysis, so it enters the mitochondria. This is pyruvate in the mitochondria. So we can run two separate reactions. So remember, we can go pyruvate to acetyl-CoA and make the pyruvate dehydrogenase complex, or run it in the pyruvate dehydrogenase complex. Or we can also run the pyruvate carboxylase and make oxaloacetate. So pyruvate can be converted into those two different things. So this shouldn't be new. We talked about this in gluconeogenesis and TCA. Okay, so now our first enzyme in TCA is citrate synthase. What it does, it takes acetyl-CoA and oxaloacetate, and it converts it into citrate. Okay, so the reason we convert to citrate when we're running fatty acids is because this acetyl-CoA cannot cross the membrane. So we need to convert it into something else. So we convert it to citrate, and then citrate goes into this transporter, and it's called the tricarboxylate transport system. This is important because you might be asked about this, how the cell, what transporter does the cell use when it's when it's making fatty acids? So it's called the tricarboxylate transport system. This takes citrate and sends it into the cytosol. We have this enzyme, it's called ATP citrate lyase. Okay, so this is the opposite of citrate synthase. It uses ATP, and this is one reason why the cell needs a lot of ATP and energy when we're running fatty acid synthesis. Takes citrate and then breaks it into acetyl-CoA. So this is acetyl-CoA, and then we also break it into R-oxaloacetate. Okay, So this now can start fatty acids. We have acetyl-CoA in the cytosol, so we can start making our fatty acids. Now let's talk about the other part of the shuttle to continue to run it. We run malate dehydrogenase, so we convert oxaloacetate back into malate. So this is the enzyme from TCA. This shouldn't be new stuff. Okay, so now we have a new enzyme. It takes malate and converts it to pyruvate. So this is called malic enzyme. This enzyme is important because it makes NADPH in the process. NADP gets reduced to NADPH, and then malate gets oxidized to pyruvate. This NADPH supplies half of the NADPH the cell uses. So this is important to run the second part of the shuttle so we can make more NADPH to the cell. And then pyruvate is made, and then pyruvate can continue this way again. So we make acetyl-CoA, and then we convert it back, and then we keep running this cycle, making more acetyl-CoA and more NADPH, convert back to pyruvate, pyruvate can then enter. So like I said, ATP citrate lyase, so that makes oxaloacetate and acetyl-CoA. Oxaloacetate is reduced to malate to continue the shuttle, so this is the malate dehydrogenase. And then malic enzyme produces half of the NADPH needed for fatty acid synthesis, and in the process we make pyruvate to continue the shuttle to run again. Okay, it's pretty simple and straightforward, but it might be a little hard to memorize the different substrates and where they occur. So this is the mitochondria and this is the cytosol. Make sure you keep track of what reactions are running to what direction in different locations in the cell. Okay, so we made our acetyl-CoA. Now we can start making fatty acids. So the first enzyme, it's the rate limiting and the committed step for fatty acid synthesis. So as soon as we run this enzyme, we're making fatty acids. So this is done by acetyl-CoA carboxylase, or it's called ACC. We'll talk about ACC1 and ACC2. The one that starts fatty acid synthesis is called ACC1. ACC2 has a regulatory function. So the one that starts fatty acid synthesis is the one ACC1, which we'll talk about both, but the one in the picture here is ACC1. Okay. ACC takes acetyl-CoA and converts it to malonyl-CoA. And then malonyl-CoA functions as a 2-carbon donor, and then fatty acid synthesis takes both, and then it keeps adding 2 carbons on different acetyl-CoAs until you get your fatty acid. Okay. So let's look at the acetyl-CoA carboxylase structure. So like I said, it has 2 different isoforms, ACC1 and ACC2. If you're confused on what isoforms is, remember when the cell can have different exons, and they can have the different splicing sites based on those exons. And then when you splice or attach different exons together, you get different isoforms. So that's what it means by the different isoforms. They're usually transcribed from the same gene, and then you have different splicing sites which result in different isoforms. But that's what isoforms is. And then it polymerizes into filaments, and like I said, it's a single polypeptide and it has multiple domains. Okay. So ACC1 is located in the cytosol. This is in the liver and adipose tissue, so this is the one that starts fatty acid synthesis. Okay, so this is the one we'll talk about. ACC2 is in the heart, liver, and skeletal muscle. ACC2 is the same thing. It takes acetyl-CoA and converts it to malonyl-CoA. The only difference is it has a mitochondrial targeting motif. So what this does is it targets it on the outer membrane of the mitochondria. And what this does is, when we'll talk about beta oxidation, we have this enzyme called CPT1, which is the main, it's the rate limiting step in beta oxidation, the opposite of fatty acid synthesis. So what happens is this ACC in the outer mitochondrial membrane, when it makes the malonyl-CoA the product of ACC2, malonyl-CoA can inhibit CPT1. So this is another way to regulate between beta oxidation and fatty acid synthesis. So I'll say it again. ACC2 makes malonyl-CoA as well, except it's in the outer mitochondrial membrane. So while the cell is running beta oxidation, if ACC2 is running, it'll make malonyl-CoA and will inhibit CPT1, which is the main enzyme in beta oxidation. Okay, so it's kind of when both ACCs are running, we want to inhibit beta oxidation to prevent any futile cycles. So that's just another way the cell regulates between those two different pathways. Okay, so let's look at the mechanism of acetyl-CoA carboxylase. So it uses biotin as a cofactor. So if we go back, we will remember the enzyme pyruvate carboxylase, which takes pyruvate and converts it into oxaloacetate. It's also really similar that it uses biotin, and the reason is because most of the carboxylase will use a biotin or cofactor. The reason is because biotin it carboxylates stuff. It transfers CO2 from different substrates. That's why whenever you see a carboxylase reaction, you'll see that the cofactor is usually biotin. Okay, so biotin takes the CO2 from bicarbonate and then recarboxylate in acetyl-CoA. The mechanism is very similar to the pyruvate carboxylase one if you're asked about it. So I would go back and it would be too long if I include it in this recording as well, but I would go back if you're confused about it. But we take CO2 from bicarbonate and then we get this carboxybiotin. It's bounded to a lysine residue in the enzyme. So this is a lysine residue in the enzyme. And then we create this biotin lysine arm that's able to transfer the CO2 to acetyl-CoA, and then it continues the reaction forward. Because we transfer CO2 to acetyl-CoA and then we make malonyl-CoA, and then that's the product of ACC. Okay, so biotin acts as a carrier and it also uses ATP in the process. So let's zoom in. So this is the lysine arm. It's in a biotin carrier protein. We have the biotin binded to the lysine arm. We hydrolyze ATP. Remember when we're adding CO2 from bicarbonate, so this is bicarbonate. We add CO2 on biotin arm, so we get the carboxybiotin. So this is now carboxybiotin. And then we have acetyl-CoA, and then we add this CO2 into acetyl-CoA, and we get malonyl-CoA. We get biotin again, which can start the reaction forward. This is a summary of the mechanism of it. If you want to know the exact things that attack each other, go back to the pyruvate carboxylase mechanism. It works very similarly. Okay, so let's look at more regulation of ACC. So we said it can be regulated by location. Fatty acid synthesis occurs in the cytosol, beta oxidation occurs in the mitochondria. And then we also said the product of ACC2 inhibits CPT1, which CPT is the enzyme in beta oxidation. So that's another way to regulate between ACC and CPT. And I'll upload a summary of different ways to start regulate between those two, like I do with when I upload enzymes and regulation. Okay, another way it can be regulated is by post-translational modification, so by phosphates in when it gets phosphorylated and dephosphorylated. So an active carboxylase, ACC, does not have a phosphate on it, and it's also in a polymerized form. It's important. And then an inactive has a phosphate and it's depolymerized. Okay, so the enzyme that deactivates ACC is called AMP activated protein kinase, and another name for it is AMPK. I'm sure you've seen AMPK by now, but if you haven't, I have a slide, we'll talk about it briefly. And we'll also talk about it when you talk about energy regulation because it's a master regulator. And then protein phosphatase 2A takes the phosphate off and it activates carboxylase. Okay, so another thing that can act on it is citrate can actually bind to the allosteric site of carboxylase, So remember, when we have a lot of citrate, it signals the cell that there's a lot of TC activity. The cell is in a high energy state. So we want ACC to be active to run fatty acid synthesis. So citrate can bind into the inactive carboxylase form, and it polymerizes it. And so that partially activates it, so then ACC is able to run forward. So that's one way that citrate can regulate ACC. And then palmitoyl-CoA, which is one of the products that can be made from fatty acid synthesis. So this induces filament disassembly, and then palmitoyl-CoA can bind here and then deactivate it. Kind of the opposite of what citrate does. So citrate can partially activate ACC because citrate is one way the cell signals that it has high energy. Okay. AMPK, so briefly, AMPK is activated by AMP. So remember we talked about the relationship between ATP and AMP. Remember, when AMP is high, ATP is low, right? So when AMP is high, that means the cell is in a low energy state, so it binds and activates AMPK, and then when we have low energy, we want to inhibit anabolic reactions and run catabolic reactions, because AMPK is activated when the cell is in a low energy state. Catabolic reactions break things down, then they give you energy. Anabolic reactions use energy and store things for later. So when we have low energy, we want to inhibit the anabolic reactions, and that's the mechanism that the cell uses. Okay, we'll briefly go over AMPK. So it's a master regulator. Like I said, it regulates a bunch of different pathways in the cell together. So it's activated by AMP, like I mentioned. So this is the low energy state when AMP levels are high. It promotes catabolic reactions and it inhibits anabolic reactions. So let's look at what it does for a second. So AMPK, so remember we want to promote catabolic reactions. So it activates this, as you don't have to know the different stuff that it activates, but it activates this protein which increases glucose uptake. It activates PFK2, which we talked about in glycolysis, and this helps upregulate glycolysis. So it inhibits ACC2, and because we said ACC2 inhibits CPT1, which is an enzyme of beta oxidation, so it inhibits the inhibitor of beta oxidation. So beta oxidation increases. So these are all catabolic reactions so far, because they make energy for the cell. We'll also upregulate mitochondrial biogenesis. You guys don't have to know this, but we upregulate mitochondrial biogenesis because in the mitochondria a lot of catabolic reactions happen, like beta oxidation, so we'll make more mitochondria. We'll inhibit ACC1, so we'll downregulate fatty acid synthesis. We'll inhibit HMGR, so this is this is the rate limiting step in cholesterol metabolism or sterol synthesis, which we'll talk about later in the semester. So we'll inhibit this as well, and then we'll inhibit glycogen synthesis. So these are all anabolic, and then we'll also inhibit this one as well is anabolic. We'll inhibit protein synthesis as well. So the ones we didn't talk about is protein. We didn't talk about cholesterol yet. I don't think we'll talk about this throughout the whole semester, but the other ones we talked about. Glycogen synthesis, fatty acid synthesis. We'll talk about beta oxidation in the next lecture. Okay. So now we made our malonyl-CoA with ACC. Our second enzyme is fatty acid synthase. So this is the metabolome, which has a different. So it's one multifunctional protein, but it has all these different proteins that are transcribed that are able to make the fatty acid. So in mammals, it's one multifunctional protein, but bacteria actually have several complexes. So that's one way to distinguish mammals between bacteria. It catalyzes the remaining reactions of fatty acid by biosynthesis. So this enzyme exists as a dimer or a monomer. So the monomer has all the enzymes the cell needs to run the reactions, but only the dimer is active. So the monomer needs to dimerize for this enzyme to function properly. Okay, so let's look at the fatty acid synthase briefly. So it has 7 catalytic sites, 7 different separate reactions. So this is the dimer over here. So this, if we break it, so we have a monomer on each side. So each of the monomer has the 7, can run the 7 separate catalytic reactions. It's organized from head to toe. So you'll see head to toe, or head to foot. My bad. No, I don't know where I got toe from. It uses this carrier protein called ACP, and I have a slide on why it uses that and why it's important. Basically, ACP, because we're running all these reactions on the same enzyme, we'll need to transfer the substrates from different catalytic sites, so ACP kind of helps with that. It's a flexible domain that shuttles substrates between all the different catalytic sites. So that's the main purpose of ACP. Okay, so ACP, let's look at the structure of it. So this is what it stands for. I'm not going to try to pronounce that, or I could. Phosphopentathene prostatic group, so that's what it... oh wait, that's just a group of ACP. I'm sorry. And then this group has a cysteamine, and what the cysteamine does is it has this thioester bond, which very similar to the one you make in the pyruvate dehydrogenase complex. Remember, this is high energy. So this bond is important because we make this high energy bond that's able to be broken down and gives this, it's a high energy bond that's be able to broken down and gives the cell a bunch of energy. Or it makes it really easy to break it down, I guess. So the 2 goals of it is to make this thioester bond with an acyl group, and then this long arm, so you'll see, it's actually pretty long, like in the cell, I think it's like 2 nanometers, no, not 2, is it 2? Yeah, it's 2 nanometers, something like that. But it's a long arm, and it's able to access all the separate active sites in the cell. So it swings back and forth. So this is the ACP, and this is the group that it has, and in that group it has a cysteamine group that's able to form the thioester bond. So it swings back and forth and shuttles things between the different active sites. Okay, so now we made acetyl-CoA, or we made methylene-CoA. Zoom in real quick. We make acetyl-CoA. This is the entire fatty acid synthesis. I know it's really long, and you guys. I mean, it repeats itself, so hopefully, once you don't have to memorize everything, you just memorize what happens first. And you kind of just use logic to get there. So fatty acid synthase does all those reactions. That will eventually go from acetyl-CoA and malonyl-CoA to palmitate, which is... Oh, I was right. It's a 16 carbon molecule. Is it 16? Yeah, 16. I'm going to look it up after, but I'm pretty sure it's 16. Okay, so it's we'll divide it into 3 stages. So we'll have the transfer stage, and then we'll have an elongation stage. So basically, we'll just repeat what we did in the transfer stages, and then we elongate the fatty acid more, and then we'll have a termination state, and then that will release our palmitate, or whatever fatty acid we're trying to make. Okay, so let's look at the transfer state briefly. I don't like this, so this is a better view that I'll talk about, but let's look briefly over this. So we have acetyl-CoA and then malonyl-CoA. Okay, so we make malonyl-CoA from acetyl-CoA, so this is, remember, ACC1. We'll need both to start the first reaction. Okay, so this enzyme called MAT, it takes acetyl-CoA and attaches it into the ACP site of fatty acid synthase. So it attaches it to ACP, and then the next 2 reactions kind of happen at the same time. This enzyme KS, so we had MAT, the first one. KS takes acetyl-CoA and it transfers it to a sulfohydryl site in the enzyme. And in that process, the ACP site frees. So then malonyl-CoA binds to the ACP site by MAT as well. And then malonyl-CoA attacks acetyl-CoA, and then you get your first acetyl aceto as this thing, yeah, acetoacetal ACP, which is the first product of the transfer stage. Okay, so I don't like that view. I like this one more. Uh, this is easier to understand. So this is the fatty acid synthase. We have the two different sites. We have the KS and the ACP site. So this is just another way to look at like what we talked about here. MAT takes acetyl-CoA. We take the CoA and S out, and we attach it on the ACP site. And then KS takes this acetyl-CoA, and then we put it on this sulfohydryl group next to KS. Okay, so when we do that, the ACP site frees. MAT then uses ATP. So this is one of the steps that uses ATP, and then we attach. Oh wait, I'm sorry. This is not one of the steps that uses ATP. This is ACC. So malonyl CoA is made from ACC1. MAT takes malonyl CoA. We attach it on the ACP site. So now we have malonyl CoA here, and then we have acetyl-CoA on the KS site. So malonyl CoA is on the ACP site, and then acetyl-CoA is on the KS site. And then the next reaction is catalyzed by KS. Basically, malonyl CoA attacks acetyl-CoA. We get a decarboxylation, and then we're left with the acetoacetal ACP. This is acetoacetal ACP. So what did we accomplish here? So we had an acetyl-CoA molecule, so it's a 2-carbon molecule, and then we attached another 2 carbons. So we have 1, 2, 3, 4. So we had a 2-carbon acetyl-CoA, and then we used the malonyl CoA as a 2-carbon donor to make a 4-carbon molecule. So that's kind of how this fatty acid synthesis run. The transfer stage transfers the 2 carbons, and then we'll have the elongation where it repeats that process forward. Okay, so and then you have this Kr, but I'll briefly talk about Kr afterwards. So acetyl is transferred to ACP by MAT, so this is just a summary of everything we went over. Then the acetyl is transferred to a sulfohydryl group of KS. Malonyl is added to ACP by MAT, and then acetyl is added to malonyl by KS. So this is the step right here. In that process, CO2 is lost, and then this happens when the carboanion. So we make this carboanion here on malonyl CoA, and it attacks acetyl-CoA. Mechanism of this is not as necessary as knowing what's going on. Okay, so now we make our acetoacetal ACP. Kr comes in, will reduce, and then this is the first enzyme that uses NADPH. All the ATP that is used by ACC1. So when we give the overall reaction, remember we said we need a lot of ATP and we need a lot of NADPH. So all the ATP is used by ACC1, and then most of the NADPH, or all of the NADPH, is used in these two reactions here. So Kr uses NADPH, and it reduces this ketone to an alcohol. Okay, so we get the D-beta hydroxybutyryl ACP when we run that reaction. And then we have DH over here. I'm not gonna try to pronounce all these enzyme names. You can just call them KR, DH, and EAR. DH, what it does is it dehydrates the substrate, and then we get this double bond. Okay, so then we end up with alpha beta transbutanol ACP, and then ER comes in. I know it's called ER, whatever. It uses NADPH again, so this is the second enzyme that uses NADPH, and then we get butyryl ACP. So after the transfer stage, and then the transfer stage is we transferred the malonyl CoA. We added a 2-carbon donor, but then we do a bunch of reduction, and then we end up with a 4-carbon molecule. So we started with this, and now we added 2 more CH3s. So you see how this can be repeated, and then we keep adding different carbons depending on what we want. Okay, so this is okay. So this is kind of this is the view I like of the transfers. So I'm sorry. So this is the butyrate ACP that we made in the last step. Okay, so we made the butyrate ACP. It's on the ACP site. So this will start the same way that we had with acetyl-CoA. So if we go back, acetyl-CoA starts on the ACP site, and then we do all these transfer reactions, and then we end up with a 4-carbon molecule over here. So this is going to be repeated with a butyl ACP, and we'll end up with a 6-carbon molecule. So KS will transfer this butyl ACP onto the KS site. This is the same stuff. ACC catalyzes this reaction from acetyl-CoA to malonyl-CoA, uses ATP. MES then takes malonyl-CoA and attaches it on the ACP site. So this is just a repetition of the transfer stage that we talked about. And then now the malonyl-CoA attacks, instead of attacking acetyl-CoA, we attack a butyl ACP, and then we have a decarboxylation. We'll end up with this, so a 6-carbon molecule on the ACP site. And then we'll do a reduction of this ketone, and then we'll just keep repeating that. Okay, so I'll go over the steps briefly. Next round starts with butyrate ACP. Just transferred to KS site by KS. Malonyl is added to the ACP site by MAT, and then butyrate is added to malonyl by KS. CO2 is lost. You make the same carbanion, but this time it attacks the butyrate. And then the double bond oxygen is then reduced, and the process is repeated. So this is what they're talking about, the double bond oxygen. Okay, so we do that 7 more times, and then you will end up with a butyl. No wait, this is the butyl ACP. We do it 7 more times. So we add 7 more 2-carbon donors, and then we'll end up with palmitoyl-CoA. Right. So we'll end up with we'll end up with palmitoyl ACP, and then this is the termination. We have palmitoyl thioesterase. So by the way, these enzymes have different names depending on what the fatty acid is. So because this is palmitoyl ACP, palmitoyl thioesterase is the one that breaks it down. So if it was, let's go back to the beginning. MAT is called malonyl acetyl-CoA ACP transcyclase, right? So when we do the second round, it's not an acetyl-CoA, it's a malonyl-CoA and a butyl ACP. So it will just have like different names, but I think you can just call them MAT, KS, KR, all of that. But just be aware of that. If you ever like see a different name instead of palmitoyl thioesterase. Okay, so that's why they call it TE, because we don't want to call it palmitoyl thioesterase because it can be something different than a palmitoyl that's being broken down. So this thioesterase breaks the ACP from the palmitate, and we get our product palmitate in the cytosol. So this is the product of acetyl-CoA. It cleaves the ACP, and we get the free palmitate. Okay, so after we get the free palmitate, we can do different things. We can modify it, but generally it's more complicated. I don't think you're expected to know them, but I'll briefly mention them. So a lot of the stuff like the trans and the cis fats, the trans and cis double bonds that come, those usually come from dietary intake. So there's an enzyme called fatty acid elongase, or EVOVL. So this can actually elongate fatty acids. So instead of palmitoyl-CoA, we can make a longer, like stearate CoA or something like that. It can elongate fatty acids. This usually happens in the ER, or sometimes in the mitochondria as well. Desaturases. So they're monooxygenases. Monooxygenases just uses I forgot what monooxygenation is. They use oxygen to reduce something, I think, or oxidize something. They use oxygen in an oxidation reduction reaction. Okay, but those are also in the ER, and they introduce cis double bonds. I'll briefly talk about desaturases. So those are the ones that can introduce cis double bonds. Desaturases have three components: the desaturase enzyme, cytochrome B5, and an NADPH cytochrome B5 reductase. In mammals, double bonds can't be added after carbon 9. So this is kind of important to think about. So most of all the double bonds that are added in our fatty acids, they're usually added before carbon number 9. Okay. So this is the enzyme that adds the cis fats, desaturases. So like I said, they're in the ER. So let's talk about how they work. Okay, so this is a sterol-CoA, which is a 24, I think it's a 24 carbon. Let me count that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. It's not 24, it's 18. I don't know where I got 24 from. Okay, basically it's an 18 carbon molecule. And then we can add this cis double bond with a desaturase. So the way it does this is because it's a monooxygenase, we use oxygen. Oxygen is oxidized to H2O, and then in that process, sterol-CoA is, or oxygen is reduced to H2O, and then sterol-CoA is oxidized to this cis double bond that we make, and then the electrons are transferred from cytochrome B5. You get a reduced cytochrome B5, and then that gives the electrons the cytochrome B5 reductase, which has FAD and FADH, and then the electrons are shuttled in here, right? Or it can run the other way around. So NADPH can be oxidized to NADP, and in that process, FAD is reduced to FADH2. And then when FADH2 is reduced back to FAD, the electrons from cytochrome B5 are given the cytochrome B5 with an iron +2, and then those electrons can then be given here to allow this reaction to happen. And then in the process, oxygen is reduced as well. Okay, so that's it's linked to NADPH and cytochrome B5 reductase as well. Okay, we have 2 more slides. Okay, so let's talk about the sterol-CoA desaturase. This is an example of a desaturase. So it can act both on sterol-CoA and palmitoyl-CoA. So this is actually elevated and the elevated levels of this correlate with obesity. So what does this tell us? This tells us that whenever you have cis double bonds in your fatty acids, this contributes something to obesity. And then, like I said, so it's a monooxidase, uses oxygen to produce double bonds, and it's on the cytoplasmic phase of the ER. And the ER is where so it's next. So you have the rough ER and the smooth ER. So this is where it faces the cytoplasmic phase of the ER. It's not, and then the ER like ships it to the Golgi and stuff like that for modifications, but desaturases just exist on the ER and not the Golgi. Okay, so that was pretty much that for fatty acid synthesis. I will upload this one, and I will try to record beta oxidation today hopefully by like 10 or 11 p.m. Okay, so let's go over some summary. So we have citrate goes into acetyl-CoA in the cytoplasm. ACC catalyzes acetyl-CoA to malonyl-CoA. Fatty acid synthase then catalyzes 7 different reactions. Our end product is palmitate, and this happens, regulation can be controlled by location. So remember, it happens in the cytosol. Beta oxidation happens in mitochondria. Can also be regulated by ACC. Remember, the product of ACC2 inhibits CBT1 of beta oxidation by citrate and ATP levels. So remember, citrate signals high energy levels, and then and it can act on the ACC, and then high ATP levels and AMP, they're usually regulated together. So high AMP levels would signal low energy, so that would activate AMPK, and then AMPK can then phosphorylate and inactivate ACC, and it can also be dephosphorylated by protein phosphatase 2A. So it can be regulated by citrate ATP levels, location, ACC, and then post-translational modification. So this is it for fatty acid synthesis. I'll upload the beta oxidation. It's helpful when you go over both around the same time, so then they're linked. They're very closely modified, especially with the chemistry and how they start and how we finish. Okay. If you have any questions, just email me. I hope you guys have a good day. Thanks for watching.